Okay, I'm going to start. Good afternoon, everyone. I see we have 11 people in attendance. It is very good to see you and to welcome you, and you are welcome to this online postgraduate event for the Division of Infection and Immunity at UCL. On your screen at the moment, I hope you can see a picture of UCL, and I am sitting in the orange building, this one here, which is the Cruciform building, which used to be the hospital many years ago. But I am right there, and that is our headquarters of our division. This is a place that you will come to know very well if you come and join us on any of our programs. So I'm Richard Milne, that's me in the screen there. I'm a professor of virology and head of the infection immunity program. And I'm joined in the meeting today by my colleague Melitza, who is pictured on the right. And I guess you might be able to see her on the screen as well.
Hi.
It's not exactly clear to me what you can see. And Melitza is our immunology teaching lead, and she is sitting in our main immunology building, which is about 20 minutes away from here at the Royal Free campus. It's a very beautiful new building that was only opened up last year. And you'll hear from her in a little while. But this is the agenda today. I'm going to start by introducing a little bit UCL, a little bit more our division, and then give you a sense of our kind of teaching ethos, what our pitch is, and what we would hope that you would get from any of our programs, actually. Excuse me. And then I will change tack and talk quite briefly, specifically about our MSc Infection and Immunity program, which I run. And then I'll hand over to Melitza, and she will talk about the MSc and the MRes programs that we have in the immunology sphere, experimental translational immunology. And then we will hopefully have plenty of time after that to answer your questions and follow up on any comments that you may have. So just to place UCL in context, it's a big university. I think it's now the biggest in the UK. These are the most recent data available, so they're a year behind. But we're just over 50,000 students, of whom about 20,000 are postgraduate taught students. That's master's programs, and about 6,000 are research, and about 25,000 undergraduate. So that's a big place, and it certainly does get crowded at times, particularly near the beginning of the academic year. And that can be quite intimidating and overwhelming. But something to remember, I mean, you are all in the business of choosing degrees at the moment, or postgraduate degrees, and you'll probably be thinking a lot about university league tables and rankings and so on and so forth. And I would just point out that those typically are associated with the whole university or a large department or faculty within it. And actually your experience is largely governed and defined by the immediate environment that you occupy, which is your program and your host department or division. UCL calls departments divisions. And the Division of Infection Immunity, which is our division, is actually quite small, and I think quite a welcoming and friendly place. And it's certainly a good place, I think, to be studying on these programs in this disciplinary area. So what's the division all about? It's a division within the Faculty of Medicine. We have about 40 principal investigators. What I mean by that is those are individuals who are scientists who run a research group and typically will be funded to deliver research in a particular area. The research that we do is pretty broad, actually, within the scope of infection and immunity, and it spans really, I think, quite genuinely the distance between what we call bench to bedside, and that refers to the laboratory bench, where the very fundamental pipetting type science is done, right through to the hospital bedside, and quite fundamental clinical research, and everything in between. And that's what you want, really. Infection immunity are disciplines of science, but also disciplines of medicine. About a third of our investigators are, in fact, medical doctors and clinician scientists. And both here in the Cruciform building, where, as I look out my window, I can see the University College Hospital, which is one of our main teaching hospitals, and also at the Royal Free Campus, where the other building, the Pears building, which is our new immunology building, is based. We're both immediately adjacent to the hospitals. So that very much colours the kind of work we do. And I think for delivering research and training in infection and immunity, which are both at this clinical scientific interface, we really couldn't be better positioned for that. And so that's what we do. We in all of these programs, the real point is to do research-informed teaching. Now, that doesn't mean that you just come and listen to a series of professors rolling up and talking about their latest experiments, although that's part of it. It means we're going to teach you about what we do, which means that the people that are teaching you really know their stuff, and they're delivering the teaching from a perspective of someone who is an active practitioner, and with the wisdom and knowledge and experience and insight that implies. That necessarily means that we don't necessarily, sorry, I'm repeating myself. That means that we don't necessarily cover a universally comprehensive curriculum. There's some stuff, particularly in the infection sphere, that we don't do because nobody here works on it. So we don't really talk about parasites, for example, because we don't have anyone that is competent to do that at a research-led level. And so that's the first key thing. The other key, I would say, attraction of the programs is that they all involve a research project, and they're real research projects. We plonk you in a research group and we expect you to produce the goods as part of an ongoing research program. We're not interested in inventing kind of mini projects that are guaranteed to work. That's not how this business of science actually plays out. And we want you to get a realistic experience. So I don't have time to go into details about all of the research we do, but I would encourage you, if you're interested in joining us, to do your own homework and have a look at our divisional webpage. And if you click on the people, actually, the research one isn't very good, but if you click on the people, it lists each of our investigators and it has a link to a profile and their publications and so on. And that will give you a good picture of the research that we do, and by extension, therefore, the kinds of themes that will crop up in our taught programs. So I want to talk, just as I said, generally for a bit. So this is relevant for all of you, regardless of which program you're interested in. So first of all, what do we expect of you? Well, I mean, the first one's obvious, right? We want you to learn, but we want you to go beyond learning. We're not in the business of just memorizing and regurgitating. We want you to really understand, to synthesize information and leverage that to enhance a real state-of-the-art level understanding. And being a postgraduate student is different, I know, and we do expect you to take responsibility for your learning. We're not going to be spoon feeding you to the degree that perhaps happens at earlier stages in your career. And on that, so the lectures and the tutorials that comprise the sort of majority of the taught component of these programs are just the start. They do not themselves contribute sufficient information on their own for you to get a distinction level mark. You can pass probably if you just memorize the stuff that you get in the lectures, but we want you to think of the taught content as pointing you in the direction for you to continue your studies independently. So each lecture is a window onto a different aspect of the field. So we expect that you will read extensively, be really proactive in your learning, seek out information. We expect that you will participate in discussions, be curious, ask questions, really engage, because that's how you get to a point where you really are at the state of the art in scientific knowledge. And there's no compromise there. You know, you're either there or you're not. And the way you get there is by doing all of this stuff. And we, not on your own, obviously, we will support you and teach you how to read and understand and critique data and so on and so forth. So I put flippantly at the bottom that the only question you're not allowed to ask is, do I need to know this for the exam? And what I mean by that is that if you are engaging with the subject matter in the way that I'm describing, you will automatically be prepared for any assessment that we will deliver. And so that question is unnecessary. You kind of have to take my word for this at the moment, but there we go. So that's what we will expect from you. What could you expect from us in a general sense? We'll definitely provide research-informed teaching, as I've already described. I hope that most of it will be world-class and pretty good. I can't guarantee you that every lecture will be life-changing and amazingly brilliant, but I think on balance, my colleagues do a pretty good job. And my general view, incidentally, is that if you are an effective, successful investigator producing high-quality science and publishing high-impact papers, you are inevitably going to be an effective communicator of science. And so part of the reason I'm really keen on this research-based teaching idea is that it exploits that reality. If I can put in front of students really smart, really effective investigators and license them to do their thing and talk about the stuff that they're good at, that's pretty much guaranteed to give a good result. So we'll train you in, if you like, I suppose the scientific method, analysis, data interpretation, critical thinking, critical analysis, and presenting data, both generated by others and in the context of your project, generated yourselves. And throughout all of that, there will be a kind of dialogue which comprises a significant part of the feedback that you will get on your work. And I think we will challenge you to think for yourself. I mean, if you're looking for a sort of year's holiday, don't come and do these programs. They're pretty demanding, and they should be demanding because they're postgraduate degree programs at a research-intense university. But they will definitely train you rigorously in the scientific process, and that will prepare you for a career in science, for sure. So what does that all mean? How does that play out in practice? I think you will be embedded in a real research community. That's particularly true once you start your project, when you go and join a group and you will be an integral part of that group. And that's, that is a very rich experience and a big part of the whole business of learning how to be a scientist as opposed to being a student of science. So I think you'll have a very stimulating academic experience, but I think it will also be challenging, as indeed is a career in science. So that is in itself, I think, good preparation. I think you will see what I mean when I say that we teach about what we do, in that the content of these programs, and we'll give you a few sort of insights into that in a minute, do very much reflect the research and clinical excellence and expertise that we have in the division. And we do change stuff around because people move on and new people arrive. And so the content within the programs can be quite dynamic for that reason. So you'll definitely encounter some pretty superstar level investigators. And again, that's where you want to be. You want to make the most of that. I think there's a broader sort of perspective here. And I suppose at some level you could say this about almost any bioscience program, but I do think that infection and immunology in particular are cross-disciplinary. And what I mean by that is, in the context of infection, I'm a virologist, but if you want to understand viruses, you've got to have a measure of cell biology knowledge. And if you want to understand viruses of multicellular organisms, which we clearly do, you have to have an understanding of how those organisms work at a systems level. For example, the immune system. And in terms of infections, if you really want to understand them and how to intervene and control them and how they're going to evolve, you also have to have a kind of population level insight. So you're going to be touching on population health, global health, epidemiology. And so our discipline is intrinsically cross-disciplinary. And I would argue that it's also universal. And in fact, I like to sort of provoke our medical students by telling them that in the end, all disease is inflammatory in origin. And I challenge you to come up with an exception to that. So really, the discipline we work in does provide a very broad range of opportunities to access biomedicine, the whole business of biomedicine and bioscience. You can't actually cover it all, but it does give you an opportunity. I guess what I'm saying is it's not niche. Sometimes these quite focused programs are perceived as being quite niche, and that's not the case. I don't know whether any of you are interested in the part-time or flexible study, but I just want to mention that briefly. This is only relevant for the MSc programs. The MRes is not available, but we have a part-time delivery where the program is split over 2 years. The split is to be negotiated. It's not fixed, but typically you would do the core content over the 2 years and split the optional modules over 2 years. But what usually happens is that the program lead will meet with the part-time students early on and come up with a plan. The only stipulation with all of these modes is that individual modules have to be completed in a single academic year. And then we have the flexible mode where you can split the thing over up to 5 years. And again, that is to be agreed and negotiated with the program leads. So this, I'm quite supportive of this because it does open up our programs to, for example, people who are in work and for whom this might be a skill enhancing experience. How do you apply? Go to the prospectus, the online prospectus, and everything you need is there. What we're looking for in general for all of the programs is a strong 2-1 as a minimum in a relevant subject, bioscience, biology, biochemistry, that sort of thing, or a medical degree from a recognized university. It is a demanding program, and we have set the entry requirements quite high because in the end, a certain level of expertise and competence is required in order to flourish. And what we don't want to do is give you a place and then have you come here and struggle. We'd also like to see a personal statement that evidences your interest and particularly your motivation for doing the program, or whichever program you're applying for, whether that is to pursue a career in research or to further your discipline-specific knowledge, whatever, make that clear. Demonstrate to us that you're not sort of just a tourist wanting to do an MSc because it's next. We don't, for the MSc programs, we do not need you to have lab experience. That really is defined by your undergraduate program. Some people can have lab experience, some just can't. So that's not a deal breaker. We do need you to have some laboratory or research experience for the MRes. And I say laboratory or research because it is possible that you may have computational bioinformatic type research experience. That would be potentially acceptable. If there's any doubt on that, Milica can clarify, and she does engage with applicants individually if that's necessary. So that's the application. The fees are up there. I don't know if they're expensive or cheap or whether it's value for money. I would say it is, but I think that's something you should think carefully about. So what can you do when you graduate? I think, without being flippant, that you can do almost anything. The reason I say that is because I think scientific thinking, the whole business of how do you do discovery science, is really transferable, or the skills that comprise that. Not the pipetting and the running gels, but the intellectual skills, critical analysis, data interpretation, taking complex data sets and coming up with a unifying, clarifying explanation, and being able to communicate that in a compelling way on paper or through the spoken word. And creativity and original thinking, all of these things, if you think about them abstracted from the lab, would make you a really good applicant for many, many different careers. I always give the example of one of our graduates rolling up for an interview at a law firm and saying, I want to be a lawyer. And they say, well, what the hell are you doing here? You've got a degree in infection and immunity. And you say, well, actually, listen to my skills. And they'll be saying, hey, we've got to get more infection graduates. These are perfect. So the question really is, what do you want to do? And which type of program will best equip you to do that? So I'd encourage you to start thinking about this. It's really not too early to think about it. Where do our students end up? A good number end up on PhDs, either immediately or after a period of working. A good number end up as, I guess I've just captured that, working as research assistants in labs, to get a bit more of the flavor, or to maybe hone their sense of direction and what they want to do a PhD in. A subset of students each year apply for graduate entry to medical school, or at least to go to medical school as graduates, and usually are successful in that. And then, of course, there's the sort of so-called allied professions. So ways to use your scientific degree without actually doing pipetting, science writing, marketing, the whole pharma biotech operation, consultancy, clinical trials. There's a huge sort of not pseudoscience, that's terribly critical, science allied complex that you can choose from. And I say again, you keep in mind that the scientific method, which I guess many of you are already sort of familiar with, is a really robust and very transferable skill set. One other general point for our programs, it is not a requirement for you to have hepatitis B vaccine to come here, but I certainly recommend it. And in fact, if you are thinking of embarking on a career in bioscience research, you should definitely get the hepatitis B vaccine. If you've not had the full course of vaccine and the serocheck that demonstrates a robust antibody response, it is possible that there will be one or two lab projects that you're not able to do. Principally those that are either working in labs that work on hepatitis B, or possibly those that involve handling unscreened human material. So that's something to think about. The last thing I would say generically is that, you know, we are in London. The clue is in the name of the university, and London is an amazing place to be. We're right in the centre. Where I'm sitting here, almost all of the sort of famous sites of London are within about half an hour's walk. And it's a very walkable city, and it's just a great place to spend a year. And I encourage students to, you know, make the most of that. Go to the museums, go to the galleries. They're all free. It is one of the great cities of the world. So it's a fun place to hang out for a year. Right, so that's the general pitch. I want to now spend a short time just giving you some specifics for the infection and immunity program, and then I'll hand over to Melitza to do the same for the immunology programs. These pictures, incidentally, when we set up I decided that we needed to have a sort of visual way of distinguishing which program we were talking about in these online things. So these pictures for the I&I program are actually paintings done by primary school kids in this building downstairs in some of the teaching labs. We have them visit and do experiments, and we got them to draw how they envisaged microorganisms. And so this was the result. And they're quite good, I think. This one on the left has got vesicles, and this one's got sort of spike proteins on the surface. So I think they're very colourful. So this, if you come to do the infection immunity program, this will be your life whilst you're here. The first term, so October to December, it's the same stuff. It's core. There are 5 modules, and they really are broad. And that, I think, is probably the main USP of this program. It's the breadth. We cover bacteriology, virology, immunology, epidemiology. There aren't many of our sort of competitor programs that offer that breadth. Then in the second term, we do depth, and you choose 3 optional modules from a choice that I'll show you in a minute. Once they're done, I should have said that the first term content is assessed in where there are exams. They are assessed early in the beginning of the new year in January. Then you get those out of the way, do the specialist modules in the second term. Those are examined where there are exams around Easter. So by the end of the Easter break, all of the taught material is out of the way, and you come back from the Easter break and start the research project, which is full-time all the way through until the end of August, beginning of September. So each of these modules, barring the research project, counts for 15 credits, and the project is 60, and so the total adds up to 180, which is the MSc. That's what it looks like on a sort of plan basis. So first term is core modules. We have a little sort of intro to stats and numeracy, which is really an adjunct to the data interpretation module. And I should have said that that module actually runs through both terms, because we think that is the most important in the end of all of the content. And then we have the optional modules here and the project here. The core modules pretty much do what they say on the tin. The only thing to say is that the bacteriology module is partly a practical module. It's not high level. It's really to accommodate the fact that we have people with varying degrees of experience. Most people who come have done sort of lab practicals, but not everyone has done recent lab experience. So it's a way of just reminding everyone how to which end of the pipette is the business end. And then the other 3 subject-based modules are really introductory. Epidemiology. Most of our students have not done epidemiology before. Some of our students have not done virology before. Everyone's done some immunology. So it's a core, broad introductory term. The data interpretation module is all about reading papers, discussing them, critiquing them, presenting them, making an argument, and it is assessed along the same lines as that. This is the current list of optional modules. It might change slightly, but not significantly. I should just say, this is one of these sort of legal footnotes, that you can't do all of the combinations because there are more modules than there are slots in the timetable. But we organize the timetable so that the least frequently, historically chosen pairings go together. So most people seem to be reasonably happy. And just to emphasize, I'm not going to do this for every module, but just to emphasize what I mean about the research informed teaching, I'll just mention this module, which is Frontiers in TB. And this takes advantage of the fact that affiliated with our division is the Africa Health Research Institute, which is a really top-level research center in South Africa. It has two campuses, a campus in Durban and a campus in rural KwaZulu-Natal. It's very, very strong in TB and HIV, not surprisingly, given its location, both basic science and epidemiology. And as South Africa is in more or less the same time zone as the UK, or only an hour out, we thought that we would set up a module that is run jointly by UCL and ARI, the Africa Health Institute. And so this module is part in person from UCL investigators, part live online delivered by TB investigators, both from ARI and actually around the world. So that evidences my sort of comment about really genuine research informed teaching. It's a pretty cool module, actually. I've attended some of the sessions and they're amazing. The other really important part of all of our programs is the research project. And I put here what's the purpose of the project. And what I meant by that is to say that the purpose of the project is not necessarily for you to get a Nature paper. It's much more about immersing you in a top-level scientific discovery science environment and giving you the opportunity to find out firsthand what that entails in all respects. And that will tell you for sure if you want to do this or not. We talk individually to students about their interests and place them in the labs or the research groups based on that. And that works pretty well. And I've put the dates for this current year. So our current batch of students will start in the penultimate week of April and run through to August. And then they do a write-up and give a talk in a symposium and have a short oral examination. This is the pass mark for all of the degrees, actually, 50%. To get a merit, you need to get 60%, and to get a distinction, you need to get 70%. There is some small print around that, as you can imagine, but we don't need to go into that today. And that is the results for the last, 9 years. So if you set aside the COVID disruption, it's pretty consistent. Most people get merits, some people get distinctions. And I think that's how it should be. Okay, that's all I'm going to say about the infection immunity side. So I'm going to hand over to Melitza, who's going to replicate almost entirely what I've just said, but
I'll try not to.
for the immunology program. So I'm going to stop. It's all yours.
Okay, let me share my screen. Tell me that.
That's it.
You can see.
Good to go.
Excellent. So, hi everyone. As Richard already introduced me, I'm Melitza Bukmanovic-Stage. I lead the MSc and MRes in experimental translational immunology, and helped by Professor Lucy Walker, who is involved in the MRes program, especially the MRes projects. I'm not going to repeat everything Richard said, because most of the general stuff is the same for both programs. So I think I'll just take this opportunity to take you through some of the nitty-gritty detail of what these programs involve, and then also to give you the opportunity to ask the questions that you have. So let me just see if this is working properly. Right, so most of our immunology labs are in this new building, and that's where I am actually at the moment, called the Pears building, which is in Belsize Park, only probably about 20 minutes away from the Cruciform, where the main UCL campus is. It's a beautiful building. We have lots of labs on the first 2 floors, well, actually on the 3 floors, and then there's some accommodation for patients involved in clinical trials on the top. And it's one of the sort of big research institutes in Europe that is kind of created specifically for translational research. If you are interested in immunology, it's very, very likely you will end up, actually from both programs, you will end up doing your project in these labs. So the program structure, and bearing in mind that there is the MSc and the MRes, I'm going to talk about the MSc first. You have a research skills and data interpretation module, which runs across 2 terms. This is a key module. As Richard mentioned, it's very similar. We spend a lot of time going through papers, discussing data, learning how to interpret and critique other people's data. And this is a great preparation for your research project and obviously for your future careers. Also involves a stats component. In term 1, you do cellular pathology and also a fundamental immunology kind of course. This is a large module, the idea being to bring everyone sort of up to the same level, bearing in mind that people join this course from quite different backgrounds. It starts sort of with a basic fundamental immunology, but it very quickly picks up and actually ends up, people talk about their research and kind of very cutting edge stuff. And again, we talk mostly about things that we are actually involved in research-wise rather than general immunology textbook knowledge. And then in term 2, you have 1 core module, which we call research topics in immunology. And again, the whole purpose of this module is to get the students involved in research that's currently going on in our division. So you would have seminars from a selection of PIs within the division. They would talk about their current research and their future interests and where this research might be going, which direction, what are the still open questions. And the students then divide themselves up into groups to create small grant proposals based on one of those research areas. So you choose what area of interest, whether that's CAR T cells or thymic development, and then you and your team work on a proposal on how you would take this work forward. And then you have optional modules, and then in term 3, so after Easter, once all the exams are done, you're engaged in the research project full time. So that's the MSc. So the MRes is somewhat different. Basically, you spend a significant amount of time in the lab doing the research project. You start about mid-November, and initially that would be part-time. About 50% of your time would be spent embedded in the research lab. And then from, again, after Easter, at the end of April, it would become a full-time project. However, the students still do the same, and we treat you as 1 cohort. So you're doing the same module, research skills, data interpretation. You're doing the immunology module, and you have 1 optional module in term 2 that you can choose. Okay. And as Richard pointed out, the MRes can only be done on a full-time basis. Okay. So this just sort of summarizes what these modules cover, but I don't think that's necessarily the best use of our time, and I have mentioned this already. I'd rather maybe give you a list of our optional modules, just so you have an idea. They're all generally related to particular areas of immunology. For MRes students, this is an optional, and for MSc students, this is a core module. And again, just a caveat, there are some modules that might not run potentially in every year, or not all combinations may be possible. But in fact, there is only 1 combination here that isn't possible, and that's partly because there is an overlap in content as well. So you would choose whatever interests you most and focus your term 2 on those modules. Right, so I think for a lot of people, and you will ask me questions, I hope, if this is something you're unclear about. I think for a lot of people who are interested in these 2 programs, it's the decision between the MSc and the MRes. That's a kind of the first decision that you want to make. So I've got this slide, which basically talks a little bit about the differences. So the key difference is the amount of time and also credit weight that is given to the research project. So MSc students start at the end of April, spend 4 months in the lab, have the symposium and a viva in September. So it's 4 months full time. The write-up is 5,000 words. There is a short oral exam at the end of sort of sometime early September. And the total credit value is 60. And remember, the total credit value of the MSc is 180. So it's a third of your credits are from this project. And certainly this is the key part of your program, I would say. Now, the MRes start much earlier. Initially, it's not full-time, it's 50-50 with taught modules. Finish at the same time, write a much longer report, have a slightly longer oral exam to discuss their work, I should say. And this MRes project is substantially higher credit value. So it's 105 credits out of the total of 180. So it is really the biggest component of the program, okay? And when you're thinking about it, what I think you should consider is partly whether you're eligible for the MRes. Do you have some relevant laboratory experience? And please, when you are applying, if you apply, do make this very clear and explicit. So what is it that you did? How much time did you actually spend doing it? Is it a practical, or were you actually involved in a research project? That is the sort of questions that I will be asking you if you haven't actually included it already in your application. I think it's also important to highlight that you will have fairly limited access to modules. You are only going to be doing one optional module. So if you haven't done much immunology in your undergraduate degree, even though you are very keen on it and you think you do want to do an MRes and you want to go into research, this is something to consider. You will not have the option to do both cancer and autoimmunity and immune deficiency. You will have to pick one. So maybe if you have only done a little bit of immunology, maybe the MSc is a better choice. Okay, so that is, again, something for you to think about. I'm very happy to discuss. Again, you can only do it full time, and we do really need to know what is it that you're interested in, because this will determine, in both cases, but in the MRes, this happens much earlier. Your personal interest will determine where your project is going to be based. So you kind of need to know that from the beginning if you are interested in the MRes. Okay, so what you can expect, I guess Richard has spoken about this. We really want you to feel part of the division. You are definitely going to be embedded in those labs. You're going to spend a lot of time talking to people. We want to encourage you to come to the seminars, to just chat to people, you know, as you meet them. And just to make the most of this opportunity to be with active top class researchers. I've just got this just because I think it's sometimes helpful to see what the timetable looks like, but bearing in mind that there is always extra reading and online content to also be covered. Especially, for example, this module is a mixture of online and sort of, I don't know, 4 hours of lectures in a week, plus about 2 or 3 hours recording. So it looks, I know this is term 1 for our students. It does look a little bit light, but believe me, by about November, everyone says, oh my God, there is so much work to do. So it is not light at all. You do really need to think, you know, you are going to be busy if you come to any of these programs. And this is potentially what term 2 might look like. So depending on what you choose, most of our modules have 2-hour slots a week. So 4 hours in total, spread around across the week. And these, very briefly, because these programs are new, we've only started these programs when the new pairs building was opened, and that was slightly delayed by COVID. So these are our current kind of last 3 years of data that we have on our students. And we had, you know, a mixture of merit and distinctions. And we have some data on where our students go. A lot of our MRes students are now on PhD programs, or they're doing, and the same actually for the MSc. If they're not on PhD programs, they're working as research assistant, applying for PhD programs or making a decision whether that is something they want to pursue, or they have gone off to do various other sort of related careers. I think I'll stop there and I will stop sharing. and we're going to look at the Q&A. I hope you have posted some questions.
Okay. Thanks very much, Melissa.
Okay.
Right. So you are now welcome to post your questions in the Q&A segment of Zoom, and we will answer them, or we will try to answer them. Okay. So Naisha says, for the MSc in infection and immunity, once we are assigned to a specific group, do we get to do our original research or continue to work on a preset research topic? I'm not quite sure I understand what you're asking.
I understand the question, actually. I think I understand the question.
Okay.
But hopefully we'll get corrected. I think the question is the research project a set topic, or is there kind of, they do their own original research? And I think, I don't know if I've got this right. Can you let us know, Naisha? Is that what you were thinking?
Well, let me explain. I mean, let me explain how it works.
Explain the process, and I think that will answer the question.
For both these programs, actually, well, all of them.
For all of them, really.
But you ask for the I&I within the first sort of few days, well, not first few days, first few weeks of the first term, I meet individually with each of you for a preliminary chat, but we then have a proper conversation in the second term about your research interests. And we also get you to give us some information sort of on a spreadsheet. And then having then met all of the students on the program, I then, because I know all the labs and what everyone is doing, I know where you will each best fit. So I direct you to the lab that I think is the best fit for you, and you go there, and then you have a conversation with the lab head about what is available and what the project or projects are that might be done. They will be real projects. Most frequently, they would be a sort of sub-project of, let's say, a PhD student's work or a postdoc's work. So you might have a particular assay to develop or a particular type of question to answer, but it will be something that is embedded in the ongoing research program that lab or that research group is delivering. But there's no, you're not upfront choosing a particular sort of topic. It doesn't work like that. And in fact, what you'll come to realize is that research doesn't work like that. So I hope that has answered your question. The reason we do it this way, rather than just giving you a list of titles, which is the more common way, is because my perspective is that there's no way you can possibly make an informed choice about a project from a one-sentence title, or probably even from an abstract, because you've got no idea what of anything else that's going on there. Much better to have the discussion, get a sense of your general interests, and then send you to a lab that best fits those. So that's why we do that. And the students like it. It works pretty well. Okay, thank you, Naisha. Right, Anonymous. What is the average acceptance rate for the MRes? Thank you. I'll hand that to Melissa, but we must colour the answer here with the fact that the sample size is very small, and as future scientists, you will appreciate that one has to be very conscious about over-interpreting.
Yes, I was actually trying to find sort of the data that would actually tell us. I would say there is probably about 40, 50 applicants and we have anywhere between 8 or sort of 12 people join the program. But sometimes you might get accepted and then student might choose to go somewhere else, or they might choose to do something else or have no funding or want, so it's not necessarily about just acceptance. The other thing that sometimes happen for the MRes, people will apply and then I would judge their application not to be quite right for the MRes and decide to discuss with them if they would like to do the MSc instead. But yeah, so that sort of answers your question, I think.
Okay, thanks. So we've got actually more questions than I've ever before seen in one of these events. Well done, everyone. And we've only got 8 minutes left, so let's ramp it up a bit. If my background, so I'm going to batch up 3 questions, right? They're for you, Melissa. So number 1, if my background is biochem, mol-biol, how much immunology knowledge should I have before joining the MRes? Second, if my prior research experiences are in molecular biology and cell biology, am I still able to apply for the MRes? And that's it. Go, so, quick, 1 minute.
So very quickly, yes, research experience is very transferable. And if it's molecular cellular biology, that's not a problem, as long as you have a very clear explanation of why you're interested, what you're interested in immunology related wise. I think for how much immunology knowledge you should have, I mean, it would be good to have done at least one sort of higher level immunology module, at least during your undergrad. But that's something we can discuss offline if you wish, or... Yeah.
Okay. For the MSc Infection Immunity, which building would most of the lectures or modules take place? That is an unanswerable question. I can tell you that in terms of the teaching, it is almost entirely delivered in the Bloomsbury campus of UCL.
As is actually for the immunology program, just
yes, all across the board. But in terms of buildings and rooms, I don't know at this point because it's all organised centrally. For the MSc Infection Immunity, are there certain projects international students cannot do? No, not that I'm aware of. As long as you're here, you are treated no differently from any other student. Naisha, again, are we assigned to classes when we get to the program, or do we have to register separately? Also, are we assigned special modules, or do we get to choose one? And is it competitive? You don't have to do anything in advance. When you get here, the first term is all core anyway. You don't have to choose anything, so it doesn't matter. You have time to choose the term 2 options, and students on programs within our division, like these ones, have priority for all of our modules. So I'm not aware that we have ever rejected one of our own students from one of our own modules. So I think I can reassure you on that. With a master's, with a master's alone, what can you do career-wise? If you then did a PhD, what other career opportunities would it open up for you? Generally, if you have a master's, you are equipped to work as a research assistant, a research technician. I'm talking lab-wise. All of those allied professions, I think it would be true there as well. The one thing that you will not ultimately be able to do in this field, at least, without a PhD, is go down the academic career track and apply for grants, become a professor, and so on and so forth. But by the time, if you do a master's, by the time you decide that you're interested in doing that, you will definitely have decided whether or not you want to do a PhD. So I think a PhD is not necessary for many of those careers that I have mentioned, although it probably does enhance your marketability. It is necessary for pursuing science as a researcher, a lead researcher in the academic sphere. Pharma is a bit different. It's somewhat more based about experience, and I can't speak directly to the recruitment criteria there. Right, I'm trying to get through all of the questions I can.
On the MRes project, so I think...
Yeah, so in terms of the MRes project, how do we choose our project? Is it possible to get in touch with the PI prior to the start of the semester if I'm particularly interested in their research group?
And there is a linked question to that later, but let me so
let me just read the other one. What will there be an opportunity to discuss allocation before beginning the course? What's the process?
Okay, so these are very linked. The process of project allocation is very similar to what Richard described. We will ask all the MRes and the MSc students to tell us about their interests, and we will then meet them individually and discuss how their interests fit in with the research that's currently going on. So, and then we would suggest the lab that we think fits with their interests. you can't really approach PIs and arrange projects separately from that process. And certainly there is no point in trying to think before you have got a place what project you might want to do, partly because we don't necessarily know year on year which projects will be available. So to discuss with a PI currently that you would really like a project on, I don't know, NK cells
I would just add one thing to that. It's not really a competitive process, and it's a mistake to come here with your heart set on doing a project on CDX, on cell type Y, because that's not quite the name of the game. It's about giving you a rich, broad, accurate research experience that prepares you to do that as a career. If you are absolutely hell-bent on working on CDX on cell type Y, then you probably should be thinking more about applying directly for a PhD in a appropriate lab. Right, what's the acceptance rate for the I&I MSc? So far this year, in the applications I've processed so far, it's been about 25%, so 1 in 4. Does going into research provide a stable career? Are you likely always to have a job? My goodness. I think it depends what you do. I think there's a lot of talk about the precarious nature of research. That is really talking about academic research paths. And the problem there is that the pyramid is very flat. In other words, there are lots of people getting PhDs. Many of them go on to do postdoctoral research, but then there is a big cutoff point because there are not as many jobs or positions to do academic research, i.e. sort of lecturer type professor jobs, as there are postdocs. I think you certainly can use this as the basis for a stable career. I would just say that stable does not necessarily mean constant and unvarying. I think more and more we're seeing kind of portfolio careers where you do move around. So I think gone are the days when you get your PhD, get a job in a university, and then retire with your carriage clock 40 years later. I think it's more dynamic than that. But I think certainly in this business, in this field, infection and immunity, it's very well funded. It's very current. It's right at the heart of innovation in medical science. I don't think there's ever going to be a shortage of questions to answer or funding available for competent researchers doing high impact projects. So I would be positive, but I think do your own homework, do your due diligence, work out what you want to do, and then consider if the career is something that can sustain that. It's certainly not for everyone. But I think if you've come to this and you've got this far along thinking around this, then you should give it a go. That would be my advice. I mean, discovery, which is the job of a scientist, is amazing. There's no other career that offers that. So I think that's a good way to leave this.
Shall we answer about the deadline for applications? I think that might be. So the deadline.
That's all on the prospectus. It's all very clear on the prospectus.
And so yes, different whether you need a visa or not, but it's all on the page, so you'll be able to.
I think it's April for. No, if you need a visa, and October if you, or September if you don't.
Yeah, end of August.
Okay.
Yeah.
All right. I think we're done. I'm not sure if I have to stop this. Anyway, thank you all for coming. I wish you well in your search for positions. I'm going to click